We're live. Hey, everybody, Dr. Rob Silverman here. I've got my favorite, most intelligent returning guest, Dr. Brent Dorval. I needed him to come on. We've had so many questions discussing, the health pandemic, virus, COVID-19, and there's some new breaking news. So I'm going to bring Dr. Dorval in and say, hey, how are you? How are things going?
I'm very well. Thank you, Dr. Silverman. Appreciate you having me on this morning. And good morning to everybody else that's listening or viewing in. Today, we have an interesting topic, continuation of an interesting topic, let's say. We're going to talk a little bit of just a quick review about coronavirus, and then some things that we're doing at KBMO that will probably be of interest to not only practitioners out there, but maybe even a lot of patients as well. So just the recap, KBMO is a diagnostic company. We do a food sensitivity test. I think everybody's aware of that. We're going to focus more or less on coronavirus today. And we have a PCR test that's specific for SARS-CoV-2. It detects the initial mRNA, and it doesn't cross-react with those other common strains that are out there, the other common flu strains, OC43, 229E, MERS, SARS, and a few others. So we have that, and that's an early detection test for mRNA of coronavirus. And you would use that very early on in infection, let's say 3 to 5 days or so after somebody's been exposed or shows the first symptom, in particular a fever. The next thing we'd like to address is previously we had a nice rapid test that was, and the PCR is EUA approved, by the way, or authorized, not approved. And previously we talked about the detecting the N protein or the nuclear protein on a test that we had that is also EUA authorized. And it's a very, very good test. Again, doesn't show cross-reactivity with common circulating viruses. It does react specifically with COVID-2. Now, we have another interesting test that's just come on board, and it detects not only the nuclear protein, which is an internal protein that's associated with the RNA, packaging. We have a test that detects both the N protein as well as the surface spike protein or the S protein. And that's very, very important. It detects antibodies against this protein. And this is the protein that binds to the ACE2 receptor on type 2 lung cells and other cells and initiates infection. So we have a test that detects antibody to both simultaneously, and it detects very early infection and no cross-reactivity, very important. What we found in the context of using this test is that people that have been vaccinated with the Mrna or the Pfizer vaccine show a very high titer S antibody response, and we can detect it using this rapid test. And it's not like it's a surprising development or anything particularly earth-shattering, but when we first started seeing this, we thought that practitioners and patients would be interested because, sure, you get your 2 shots of the, let's say just the Mrna, you shot 1 and shot 2. 9 to 10 days after the second shot, you show a strong antibody response against the S protein. So it kind of confirms in a way that the vaccination has worked, because if I test myself with the N for the N protein alone, I'm negative. If I test for the S in the N protein, I'm a strong positive. And as we all know, the Mrna and the Pfizer vaccine, and also the Johnson & Johnson vaccine, which is coming online, they carry the code mRNA for the S protein. So after vaccination, you shouldn't be positive for the N protein unless you had an active infection, but you should be positive for the S protein. Now, we're not saying it's a neutralizing antibody. All we're saying is post-vaccination, about 9-10 days, we're seeing a strong positive response for the S protein that's coded by these vaccines, which demonstrates that you have an active B cell response and it produces IgG antibodies.
Interesting. So again, always great leading edge information. Let me unpack it a little bit. So the COVID-19 virus, if you were to be infected, it's 3 proteins, correct? It's a triple spike.
The spike protein, you'll get some response to the other proteins, but mainly it's the spike and the nuclear protein.
Now, with the mRNA viruses, which are Pfizer and Mrna,
yes,
you only get reaction to the S protein, you were saying?
That's correct, because the mRNA, it enters the cytoplasm, and then it's translated transcribed into the S protein, which then is secreted and also inserted in the membrane. You form a strong T cytotoxic cell response, but you also, when it's secreted into the extracellular compartment, you get a strong B cell response. And the B cells then generate the IgG antibody that's specific for the S protein only.
And that B cell, so everybody knows B is something that recognizes they're part of your adaptive immune system. They come in and produce the antibodies. The problem with the autoimmunity is the immature expression of B cells. You refer to it in the extrafollicular centers versus, I know, the germinal centers. So this immaturity you're not getting in the vaccine is what you're trying to say. I'm extrapolating. You're not getting that immature antibody response. So hence the decrease in theoretical autoimmunity from the vaccine versus the virus. And you guys are testing to let everybody know where their titer is, so they know how to proceed. Possibly, I'm giving you a lot here.
Yeah.
Possibly, like my dad, who's positive, but yet he's still able to go and get the shot because he wants to make sure he never gets it again.
Right.
So I just put a lot on the table for you.
Yes. Well, again, it always comes up with viruses, and this virus particular, since it's circulating, the issue of autoimmunity, and certainly antibodies that are less specific, that haven't undergone maturation, cross-react more heavily with other things in addition to the target protein, in this case, the S protein. And also in the context of some of these viruses, what happens is in coronavirus, it buds out of the endoplasmic reticulum. So what that means in a very simple way is the spike, the membrane, and the envelope proteins are inserted into the membrane of the endoplasmic reticulum, and then the RNA enters that, and then it buds off and encloses it, and then it is secreted by the Golgi. And anytime you've got the interaction of viral proteins and host proteins, in this case from the endoplasmic reticulum, you kind of set yourself up in a way for cross-reactivity. So you've got 2 things going on. One, this thing carries host membrane and probably other components. And number 2, early on in the response, you have so-called more immature or less developed antibodies, which show less specificity until you get what they call affinity maturation, which is a fancy way just to say that a successive changes in the antibody enable it to be far more specific and also to bind much more tightly to that target antigen.
Excellent. So we've got some questions. I actually had a question because somebody texted me. So you're going to see the question pop up. And you can see it. Is it possibly vaccinated and not yet build any antibodies? Should we test for antibodies in this case?
Yeah, I would say, like in medicine and science, I think anything's possible. But what they're showing with these 2 vaccines in particular, that very high 90% people are covered after vaccination. And that would indicate, so the S antibodies are neutralizing antibodies. And what they mean, what they show is that the vaccine generates a high titer of neutralizing antibodies, which prevent the virus from binding. So the antibodies bind to the S protein and then prevent binding to the ACE2 receptor and initiating infection. So what I would say is some people, and I would put it probably in the immunosuppressed category, would be more likely to show very low titer antibody, or even maybe in some cases no antibody under extreme cases. Maybe if you had a serious myeloma or something like that, you'd definitely have an altered response. So testing, again, it's more or less an individual choice for antibody. But what's nice is I had 2 doses of the Mrna vaccine. I was negative prior to vaccination. I waited about 9 days after the second shot, and I showed a strong S antibody reaction and no N protein reaction after vaccination. Now, that doesn't indicate that I have neutralizing antibodies, but what it does tell me is the vaccine has generated an IgG response, and I'm very glad to have that information, because it's like proof positive that the vaccine, at least is doing its job and producing an IgG antibody.
Well, that's good to know. So everybody can do that. So I have another question for you here. Are you working on the other found Wuhan virus? So I'm assuming they're talking about the variants.
Yes, the variants. Again, very good question. What we've shown in the approval of this test is that, and this is more recent information and really not in the package insert that was submitted to FDA, but work in the lab has shown that the UK variant, the South African variant, there's a Brazil variant, and there's also one that's coming up in California right now that's quite nasty. And while they may not give you a more serious disease, what's apparent is they're all more infectious. So that if you don't follow proper protocol, which I recommend, I still wear a mask. And I think that it's prudent until this thing is over to do that. But this test will detect those variant strains.
My chat box just went away, so I don't know where it went. So if Mrna is watching, she can come down and find it for me. That would be great. But I do know the question. The question was, everybody's asking, what's the difference in the two mRNA? So it's a twofold question. Yeah. What's the question in the twofold mRNAs? Is Pfizer and Moderna a little different? Because the reason that they're asking, I was watching a TV program, and now some pharmacies actually have one of the mRNAs in J&J. So the question is, how are they different? The two mRNAs, and how are they different than J&J?
Yeah, I mean, the mRNAs, I think they're very, very similar. And again, I don't think they are divulging their exact secret information, meaning, exactly how these mRNA constructs are synthesized from a manufacturing perspective or packaged into the liposomes. But what I can tell you about these two is they take a snippet of mRNA that has a leader and a tail sequence so that it won't be degraded in the cytoplasm. So what they do is they synthesize that mRNA sequence that codes for the S protein, and they insert that into a liposome. And what happens is when they inject this into your arm, these liposomes fuse with the membranes of your muscle cells and then initiate, once they get in the cytoplasm, they're transcribed into the protein, the S1 protein. And in both cases, one part inserts into the host membrane. That's a type 1 response, and it's T cytotoxic. The other one gets packaged via the endoplasmic reticulum and Golgi, and then it's secreted. Then antigen presenting cells and T helper cells, that's a type 2 response, MHC type 2, then generate antibodies. So those vaccines are very, very similar, whereas the Johnson & Johnson is quite different. That's inserted into an adenovirus. And what happens is that is a DNA construct. And what happens is it fuses with the membrane and then enters the cytoplasm, and then it inserts itself into the nuclear membrane and then is uncoated and is then transcribed into mRNA and then translated into the S protein. So it's a little bit more complex process for the Johnson & Johnson because it has to enter the cell membrane, then enter the nuclear membrane, then go ahead and crank out the S protein in the cytoplasm. Now, they've said that Johnson & Johnson is a little less effective. But it's all, how do you measure the endpoint and the effectiveness of a vaccine? And they're saying it's probably in the 70s or 80s. Personally, I took what was available, Moderna. I would have, without hesitation, taken Pfizer or the J&J vaccine, because what they show universally is you may have a mild infection, is what they're saying, but they'll prevent hospitalization. So you don't get the severe disease, especially the one that will take you down and you wind up on a ventilator and it's a dire circumstance from there. And you couple the vaccine with the knowledge now that the medical community has generated in terms of, now, how do we treat a COVID patient? I would say the vaccine coupled with an understanding of the treatment, a much better understanding of the treatment, the outcome, the prognosis is very, very good for people that do get exposed or do get coronavirus.
Right. You said something very good, even though it's a scientific point, it's a layman's terms. What's your endpoint? So we know that the mRNA is 2 shots. The Johnson & Johnson is 1. Johnson & Johnson doesn't need refrigeration. For areas, countries that may have transportation issues or timing issues, Johnson & Johnson is convenient. Basically, you're echoing what Dr. Fauci said. We're allowed to say his name, in that take what you can get.
Yes.
And there was a question. I'm going to put it up. Again, I'm getting some questions. Why 2 shots? So what they're asking, and I know what they mean by that, is why are you separating? And that's really going to go right to your wheelhouse.
Yeah. So again, another good question. So the first shot, will initiate the immune response, and you'll probably even get an IgG response, I would suspect, because this shot the proteins will hang around for a while. So you'll get an initial IgM in a few days, and then by day 10, 12, 15, you'll class switch to IgG antibody. So why the second shot? So the first shot, you may generate some IgG, certainly IgM. The second shot, you get the what we term is affinity maturation. Now you expose to the antigen one more time. And what happens is these B cells rearrange the coding for the IgG antibody, so it's a better fit structurally with the antigen. It's a tighter fit. It binds tighter. And it's much more effective because it's much more specific as well. So you get this tremendous maturation of the antibody response. And then these B cells, once that vaccination goes away, these B cells that have the antibodies that are affinity maturated and highly specific now will reside in the lymph nodes, in the follicles. So if they see coronavirus, a real coronavirus, the next time, within probably 24 hours, at least 24 hours, they will start cranking out a high titer, very specific antibody that goes right to the heart of the coronavirus spike and knocks that thing dead, so it will not be able to bind to the ACE2 receptor. So you get a rapid response after the second shot, and it's very specific, and it really shuts down the viral infection much more quickly.
You know, the question about autoimmunity.
Yes.
Where we get these autoantibodies. So that would be question number one. What are autoantibodies?
Okay. Autoantibodies, put simply, are antibodies that cross-react with the host cells or proteins along with the viral protein or another protein. It could be a bacteria. So you get these autoantibodies. So what happens is you get an infection by, let's just say, coronavirus. You produce an antibody, and that antibody looks and it binds to an antigen that's probably conformational, that's part host and part virus. So now you got this tremendous cross reactivity, and you end up damaging host tissue, because very often if that happens, you and we've talked about it before in the context of food sensitivity, is the antibody, IgG antibody, binds to the host tissue and viral protein simultaneously, and it will activate complement. And that's a series of proteins that once it does that, you activate C3, and it then goes to the alternative pathway, and it activates the membrane attack complex. I think again, it's C5 through C9, and it forms a pore in your host cell membrane and lyses your own tissue. So this cross-reactive antibody ends up in many cases activating complement, which causes host tissue lysis and would initiate many symptoms that are associated with organ damage, nerve damage, a variety of other tissues as well. So this cross-reactivity initiates the complement cascade.
Now, in the cross-reactivity, there's some data, there's some articles that were written by some colleagues, and they've measured it anywhere from 42 to 50% if you contract coronavirus. Mm-hmm. Whereas some of the data says a decrease, a precipitous drop in autoimmunity if you take the injection, because there's a lot of people concerned about that in vaccines. And you said something very interesting to me prior to us coming off, that the autoimmunity didn't occur at the S protein. The only opportunity for autoimmunity was at the cell membrane because of the cytoplasm, and it was an if, when, and maybe kind of deal. So could you just simplistically elaborate on that?
Yeah. Again, what happens in with many viruses, and especially this one, so I'll stick to this one, is the way that they're manufactured and how they're secreted. And this virus is one of many viruses, but it is packaged through the endoplasmic reticulum. So it carries things that are host related. And what happens is when it buds out and this virus is released, the virus is carrying S protein plus pieces of the endoplasmic reticulum, again, mostly phospholipids and things like that. But it just sets up an opportunity for the virus then to generate an immune response in the context of something that's host, and the antibodies produced against an epitope. So that's a piece of either protein or a piece of a lipid or something like that. So you produce an antibody that's, let's say, half host reactive, half antibody reactive. So the antibody is produced against that epitope, so it reacts with both host and virus simultaneously, and there's the problem. So it generates the antibody, and then the autoimmunity really is generated by or manifested by, a lot of times, activation of complement, generation of tremendous inflammation when these cells, your own host cells, are lysed.
Well, Kelly Lamb McDonald's got a question. If you've already had COVID-19, my dad, yep, why are they recommending the vaccine still? Great question.
Again, I think it's more of a booster. You want to, there may be some timing issues between having it and waiting and then getting the vaccine. You certainly want to be well on the path to recovery prior to taking the vaccine, just for obvious reasons. It does generate a fairly strong response in vaccinated people. Much so, if you get a COVID infection, and then you get the vaccine, what's been reported is that the side effects, meaning you're much more tired. You may have more inflammation at the site of injection. You may get a flu-like symptom, which is a bit stronger if you've had the disease, then got the vaccine subsequently. But what the vaccine does to the people that have already been infected, it's like a booster shot. So it just ensures one more time that your immune response is primed to the right protein, meaning the S protein, and in particular the neutralizing domain or the receptor binding domain. So that's what this vaccine does. It really focuses the immune response on the S protein, and in particular neutralizing ability of the antibody against the receptor binding domain.
So they asked this, and this is maybe a premature question, in that, have you tested anybody post J&J antibodies, and how long are they? The second question, I didn't write it up. How long are the antibodies lasting? So the J&J is first.
So that's an excellent question. No, we haven't tested anybody with J&J yet. Nobody. We're primarily from Massachusetts. We're getting that patient population, although we've tested some people out of state. But only the Pfizer and the Moderna have been generally available. So that's the population so far. But we look forward to testing J&J vaccine, because we have every expectation. I mean, if it does its job, and in the clinical trial it certainly generates S antibodies, we'll be able to detect them using this test that we have. Now, as for the length, we're not sure. But what we're going to do is take a few people, and I'll be one of the guinea pigs, and I'll be testing myself weekly using this test, just to see how long is this S antibody test going to be useful, or detect S antibodies. So I don't know right now, but I would expect, if it's like many other vaccines, it's going to be out there several months at least. And in some people, you may even approach close to maybe 10 months or a year. That would be my feeling at this point.
We got a question. Can drinking alcohol after getting vaccinated blunt the immune response, therefore decreasing efficacy?
Yeah, I mean, in general, I would say, like with any vaccine, no. Is a glass of wine going to be problematic? I'd say probably not. A lot of wine or alcohol probably is going to be an issue.
Quarters of America, I'm sorry for interrupting you, started to cheer. They're going to be quoting you. This is going to be viral now.
They'll be after me. But no, I mean, you just be prudent. I mean, anything reasonable, I think would be okay. But it does blunt the immune response, and so you just want to be careful.
Yeah. The other question, the follow up to that was, they say not to take any NSAIDs, not steroid anti-inflammatories, leave ibuprofen, Advil. Answer why, Tylenol why, and would you? It's a three-pronged question. Yeah. And would you recommend, since I know it's going to be the Tylenol, would you recommend the Tylenol before the shot?
Again, NSAIDs in general will, as you just said, will blunt the immune response. They affect both T and B cells. So and those are the active characters, so to speak, the active cells that will generate the immune response. So you want to kind of maybe tough it out a little bit after you get the vaccine if you experience some symptoms. And here's what I've heard. I mean, on my first shot, and I'll just speak from experience, it was nothing. I mean, it was just a little redness, pretty much nothing. Second shot, I had a little more soreness in my shoulder, but after a couple of days, that went away. So it didn't have a fever or anything that would, or flu-like symptoms or anything like that. I've heard of other people directly that have had, they felt a little feverish. They felt a little tired, especially after the second shot of both vaccines, by the way. But when you say, I'm protected against COVID and I won't get that, and I'll suffer through maybe a little bit of discomfort for a few days, I think people are pretty willing to suffer a little bit of discomfort for a few days, as opposed to the chance of getting coronavirus, which can be very severe. And what's coming up is very interesting. Today in the New York Times, I just read, it's going to be this discussion about vaccine passports. And this is ongoing right now throughout the world. I think personally, it's a good idea. Now, they're worried about vaccine passports availability in underdeveloped countries. You don't get as prevalent a vaccine, or in more underprivileged groups of people, as opposed to more wealthy nations or wealthy people. I think that's a legitimate concern, but I think it's a carrot and a stick. I mean, I really do think that people should be vaccinated, and I firmly believe that you should, at that point, start to almost separate out two groups of people. If you want to refuse to be vaccinated, I think that that's your right, but I don't think it's your right to expose other people that either have been vaccinated or not been vaccinated. So the stick is, you can not be vaccinated, you can choose that, but there might be a high likelihood that there'll be some countries or some airlines that may restrict your travel as a result.
So how many times should you be vaccinated once you've done it over a 3-year period? I think that's sort of a look to the future. So I think some of the concerns that people have is it's going to be a flu shot. I know this is hard to determine. Do we need a booster? Are we going to need a flu shot? Is this just another set of vaccines that we're going to have to go through? So you can take a little look in your crystal ball if you like.
Yeah, I mean, I do believe this is like going to be a flu vaccine. I think that mRNA viruses in particular mutate much more rapidly than DNA viruses that have proofreading mechanisms. So these viruses evolve quickly. They change structure of key proteins very quickly, and we're seeing that now in the variants. And it just is, the breaking point is, you get a protein now that's changed structure enough that the antibodies no longer recognize it, meaning the neutralizing antibodies. So therefore, that strain, whatever that strain is or becomes, you'll have to then make an mRNA against that particular S protein, and that will be the booster shot next year or the year after. So I do believe that this is going to become a regular shot for people on, and as yet determined basis. But if you said yearly or bi-yearly, I would say that's probably realistic.
Now, a couple of people have always asked, and I'll let you expand upon this. They were concerned that, I'm a little afraid of taking the shot because it affects the DNA. And you have said with the mRNA, it goes to the cytoplasm, never touching the nucleus.
Yes.
You say how I know there's a time frame in which that mRNA lives, and that's one of the reasons it never gets to the nucleus. So could you expand on that?
Yeah, I mean, my understanding of what's going on and how this vaccine works, at least we'll talk about Moderna and the Pfizer initially, is this S protein is produced, and some of it is escaped into the cytoplasm of the cell, and it inserts, at least a piece of it inserts with a T cell receptor, a type 1 receptor that's on all nucleated cells. So you get this little piece of S protein that now binds very tightly to the kind of co-receptor, if you will, the type 1 receptor. Now T cytotoxic cells then start to recognize that, and they say, okay, this is foreign. They expand clonally in case they see other cells that have this foreign protein with that receptor. And then these cells actually kill the cells that have been infected as if it were a real virus. So it's kind of almost a non-productive infection in that it's self-limiting. The S protein is expressed with the T cell receptor. You generate T cytotoxic cells, which then eventually kill the cells that produce the S protein, and the mRNA goes away. Now, the exact time course of that, I don't think anybody's sure, but let's say the T cytotoxic cells do the cleanup work and the cells go away over time. Now, what doesn't go away over time is when the whole protein is secreted into the extracellular space. Now you produce antibodies, those B cells and the antigen presenting cells, and also the T helper cells. So that's a type 2 response. Those now, some of them reside in tissues and vessels, but the B cells reside almost exclusively in the lymph nodes, and they're ready for another infection. So they don't kill the cell that produced the S protein, but the cytotoxic cells do. And the cytotoxic cells now, once they're done clonally expanding and doing their job, they also have T cytotoxic memory cells that also wait. So if you got infected with coronavirus, they see this protein on the cell surface, the T cytotoxic are now activated and make sure they kill that cell and really destroy the factory. So the cells are kind of the factory.
So I had another question. Why were we testing for antibodies? So, and they expanded, I just shortened it for you. Why are we testing? So if I've had COVID 10 months ago, why am I testing? And why am I testing after the vaccine? It seems like a simplistic question, but because of the test that you do at KBMO Diagnostic, people are going to want to know why the antibodies and also how do they get it?
Yeah, I mean, why you test for antibodies is basically two things. One is to confirm that you've had an infection, okay, in the case of coronavirus virus that gives you an infection. And the other part of that is once you produce IgM early, followed by IgG later, and IgG indicates that you're in the convalescent phase. So you kind of come out of the infection and you're into a convalescent phase, which is a good thing. So that's why during an active or suspected infection, you would test for antibodies. It's really kind of a confirmatory, especially if you're PCR positive. What you want to see is that patient then produce an IgM, produce an IgG, and hopefully get into the convalescent phase and be better. Now with a vaccine, and what we've noticed again at KBMO with this test is that it's just a nice way to show that if you never were infected and you received the vaccine, that you've produced an IgG response. Now, is it more a comfort? I certainly was pleased to see that. I can tell you that right now. And we just feel that we use the test for active infection, obviously, but we also thought people might be interested in getting tested to confirm that the vaccine has at least generated antibodies against the S protein. So it's not essential, but I think I was glad to know, and a lot of people that we've tested using this were very happy to see that they've had a nice IgG response.
Have you tested by chance, or are you thinking about doing a test post-antibody COVID-19 positive versus the vaccine?
Well, right now, if I understand it correctly, we're just trying to follow in certain individuals the time frame. So, as was indicated earlier, how long is this immune response really last? So we're trying to determine that in some people with multiple testing over a period of time. And it's more of a scientific question, but because what's important is once you've generated these IgG antibodies, the B cells are ready. They're in the lymph nodes. And if you get infected, they're ready within a few hours to just produce this antibody and shut this infection down quickly. So it's nice to know the length of antibody response, but the take-home message is the B cells are there, they're armed, they're just waiting for this particular virus, and if they see it, they're going to produce antibody very quickly.
So you have to excuse me, my thumbs are getting too big. I didn't get the question mark up. I apologize. I'm multitasking. So a lot of people ask, how can they get in touch with you?
Yeah, you can. I'm the Chief Scientific Officer of KBMO Diagnostics. So you can go to the website, www.kbmodiagnostics.com. And you can just call the company. You can request whatever test you're interested in, and we'll send you a draw kit or a finger stick kit. We'll get you a sample. And the nice thing that KBMO is doing, especially with the COVID test, is we're turning these tests around within 24 to 48 hours, which is absolutely critical if you have an active infection. It's less critical if you want to get a vaccine, some sort of a vaccine test. That's less critical, but we will turn this around in about 24 to 48 hours, so you're not waiting too long. And I'm always available, along with James White, who's the CEO of the company, for questions or any issues that may arise. We're very happy to help you out.
Fabulous. Well, as always, this will not be the last time that you're on.
Great.
As Oliver Wendell Holmes once said, a mind stretched by a new idea never goes to its original dimensions. Without question, you provide those adequate stretch marks. So I appreciate you coming on a Saturday, taking your time, talking about health with everybody. It's Dr. Brent Dorval, Chief Science Officer, KBMO. I'm Dr. Rob Silverman, always yours in health.
Great. A pleasure. Thank you.